Welcome to Brainwaves, the channel where we explore the fascinating world of science and technology. In today's episode, we delve into the thought-provoking topic of genetic engineering and its ethical ramifications. Genetic engineering is a groundbreaking field that involves manipulating an organism's DNA to alter its characteristics or introduce new traits. This extraordinary technology has the potential to revolutionize medicine, agriculture, and various industries. However, it also brings forth a multitude of ethical questions and concerns that must be carefully considered. Throughout this video, we will examine different aspects of the ethical ramifications associated with genetic engineering, from the potential misuse of bioweapons to the unequal access of genetic enhancements. We will discuss the risks involved in the loss of genetic diversity and the unknown long-term effects that could impact future generations. Lastly, we will explore the ethical considerations surrounding the concept of designing babies. Join us as we navigate this complex and thought-provoking subject, shedding light on the ethical challenges and dilemmas presented by genetic engineering. Welcome to the segment titled, The Potential Threat of Bioweapons. Genetic engineering, with its remarkable advancements, also raises concerns about its potential misuse in creating powerful bioweapons that could pose significant threats to humanity. Gene editing techniques, such as CRISPR, provide the ability to modify the DNA of organisms with unprecedented precision. While this offers immense potential for curing diseases and improving agricultural practices, it also opens the door to using genetic engineering for destructive purposes. Imagine a scenario where a terrorist organization or rogue nation gains access to genetic engineering technology and utilizes it to create deadly pathogens with enhanced virulence. These engineered bioweapons could spread rapidly, causing widespread illness and loss of life. The potential threat extends beyond human-engineered pathogens. Genetic engineering techniques can also be employed to modify existing pathogens found in nature, enhancing their abilities to infect, evade detection, or resist treatment. The consequences of bioweapon attacks could be devastating, with far-reaching social, economic, and geopolitical impacts. The chaotic nature of bioweapons makes them difficult to predict, detect, and counteract, posing additional challenges to global security. As we explore the ethical ramifications of genetic engineering, it is crucial to address the potential misuse of this technology for bioweapons and ensure robust safeguards, regulations, and international cooperation to prevent such catastrophic outcomes. Welcome to the section titled, Misuse of Genetic Engineering for Weaponization. Genetic engineering, a powerful tool with tremendous potential for progress, unfortunately also carries the risk of misuse for creating devastating bioweapons. The precise and targeted nature of genetic engineering enables scientists to manipulate the genetic code of organisms. This capability, while offering immense benefits for medicine and agriculture, can also be exploited to create harmful biological agents with intentional modifications. One of the primary concerns is the deliberate engineering of pathogens to enhance their virulence, transmission, or resistance to treatment. By altering specific genes, scientists can potentially create novel strains of infectious diseases that are more deadly and difficult to control. Moreover, the ease of access to genetic engineering tools and knowledge increases the likelihood of non-state actors or individuals with malicious intent exploiting this technology. The potential misuse of biological agents for terror attacks, warfare, or bioterrorism raises significant ethical dilemmas and global security challenges. It becomes crucial to implement strict regulations and oversight to prevent the diversion of genetic engineering techniques towards destructive purposes. International collaboration, monitoring biotechnology research programs, and sharing best practices are essential to minimize the risk of genetic engineering being used for weaponization. Welcome to the section titled, Implications for Humanity. The potential misuse of genetic engineering for creating bioweapons not only raises immediate security concerns but also has broader implications for the well-being and future of humanity. Firstly, the existence of powerful bioweapons created through genetic engineering threatens the safety and lives of people worldwide. These weapons could be intentionally released or accidentally unleashed, leading to widespread illness, death, and societal disruption. The fear and anxiety associated with the possibility of bioweapon attacks can have significant psychological and emotional impacts on individuals and communities. It can erode trust and destabilize societies, leading to increased social unrest and a sense of vulnerability. Furthermore, the consequences of bioweapon attacks extend beyond immediate health impacts. The healthcare systems could be overwhelmed, straining resources and making it challenging to provide adequate care to those affected. Additionally, the economic repercussions, such as loss of productivity, disruption of trade, and increased healthcare expenses, can be staggering. Moreover, the use of genetic engineering for weaponization can potentially trigger an arms race, where countries or malicious entities strive to outdo each other in developing more potent and destructive bioweapons. This race could divert resources and attention away from essential humanitarian needs, further exacerbating global inequalities. Addressing the ethical ramifications of genetic engineering requires robust international cooperation, strict regulations, and scientific transparency. By working together, we can strive for a future where the potential benefits of genetic engineering are harnessed for the betterment of humanity, while the risks of misuse are minimized and prevented. Welcome to the section titled, The Potential for Societal Inequalities. As genetic engineering becomes more widespread and accessible, there is a risk that certain enhancements and modifications will only be available to the wealthy, exacerbating existing disparities within society. Genetic enhancements, such as increased intelligence, improved physical abilities, or enhanced disease resistance, could potentially provide significant advantages to individuals who receive them. However, if these enhancements are only accessible to a privileged few, it could deepen existing inequalities and create an even wider divide between the haves and have-nots. This scenario raises concerns about social justice and fairness. If only the wealthy can afford to undergo genetic enhancements, it could perpetuate a cycle of privilege, limiting opportunities for those who cannot access these technologies. This could create a society where advantages and opportunities are unequally distributed, further marginalizing already vulnerable populations. Moreover, unequal access to genetic enhancements could lead to increased discrimination and stigmatization. Those who do not have access to certain enhancements could face societal biases or be viewed as inferior, resulting in social exclusion and a loss of dignity. It is crucial for society to address these potential inequalities by promoting equal access to genetic enhancements and ensuring that these technologies are available equitably to all. Striving for fairness and inclusivity in the distribution and use of genetic engineering advancements is essential to avoid exacerbating existing social disparities. Welcome to the section titled, Limited Access to Genetic Enhancements. As genetic engineering technologies advance and become more widely available, there is a risk that certain enhancements and modifications will only be accessible to a privileged few. One of the primary concerns is the high cost associated with genetic enhancements. The development and implementation of cutting-edge genetic technologies can be expensive, including the research, clinical trials, and specialized medical procedures involved. This cost barrier could result in limited access, as individuals without the financial means may be unable to afford these enhancements. Moreover, the monopoly of pharmaceutical companies or private clinics over genetic engineering treatments can further restrict access. If these treatments are solely available through exclusive providers, it could significantly limit who can benefit from them. This monopolistic control over genetic enhancements could lead to inflated prices and further exacerbate the inequalities in access. Limited access to genetic enhancements has implications not only on an individual level but also on societal and systemic levels. Unequal distribution of advantages can perpetuate existing social hierarchies and widen the gap between different socioeconomic groups. To ensure a more equitable future, proactive measures are necessary to address the issue of limited access. This includes advocating for affordable and accessible genetic enhancement treatments for all, promoting public research and funding, and implementing policies to prevent undue monopolization of genetic engineering technologies. Welcome to the section titled, Implications for Social Dynamics. The limited access to genetic enhancements can have far-reaching consequences for social relationships, dynamics, and the overall fabric of society. When only the wealthy have access to certain enhancements, it can reinforce existing social hierarchies and widen the gap between different socioeconomic groups. This division based on genetic advantages can lead to increased social stratification, where individuals with enhancements are perceived as superior or more valued than those without. The unequal distribution of genetic enhancements can also perpetuate discrimination and prejudices. Those without access to enhancements could face societal biases or be unfairly treated due to perceived inferiority, resulting in social exclusion and marginalization. Additionally, the growing disparities in genetic enhancements can lead to a loss of diversity and homogenization. If certain traits or characteristics are favored and widely adopted, it can diminish the richness and variety that make human societies vibrant and unique. Furthermore, the limited availability of genetic enhancements can pose challenges to social cohesion and harmony. It may create resentment and grievances among those who are denied access, leading to social tensions and potential conflicts. To prevent these negative implications, it is crucial to foster a society that values inclusivity, diversity, and equal opportunities. Promoting equitable access to genetic enhancements and challenging discriminatory attitudes and practices can help in mitigating the potential negative impact on social dynamics. Welcome to the section titled, The Importance of Genetic Diversity. Genetic diversity plays a vital role in the long-term survival and adaptability of species. It refers to the variety of genetic traits and variations within a population or species. Genetic diversity provides the building blocks for evolution and allows populations to adapt to changing environments. It enhances resilience against diseases, pests, and other environmental challenges by ensuring that a range of traits and variations are available for natural selection to act upon. Furthermore, genetic diversity facilitates the process of speciation, where new species arise over time. It enables the accumulation of genetic differences between populations and contributes to the overall biodiversity and richness of ecosystems. In addition, genetic diversity is essential for maintaining the stability and functionality of ecosystems. It supports complex ecological interactions, including nutrient cycling, pollination, and predator-prey relationships. A loss of genetic diversity can disrupt these interactions and have cascading effects on the entire ecosystem. It is crucial to preserve and protect genetic diversity to sustain healthy and resilient ecosystems. By doing so, we help ensure the long-term survival and adaptability of species in the face of environmental changes and challenges. Welcome to the section titled, Reduced Genetic Diversity Through Genetic Modifications. While genetic engineering offers the potential for targeted modifications, there is a risk of inadvertently reducing the overall genetic diversity within populations. Genetic modifications involve introducing specific genetic traits or alterations into organisms. However, the focus on these targeted modifications can lead to a narrowed gene pool within a population. By purposefully selecting and propagating certain desirable traits, other variations and genetic combinations may be lost or diminished. The reduction in genetic diversity can have negative consequences for the long-term survival and adaptability of species. With a diminished gene pool, populations become more vulnerable to environmental changes and increased susceptibility to diseases and pests. Limited genetic diversity reduces the range of options available for natural selection to act upon, potentially compromising the ability of populations to respond and adapt to evolving challenges. Furthermore, reduced genetic diversity can diminish the potential for beneficial mutations and genetic innovations to arise. These novel variations often emerge from diverse genetic backgrounds and can provide the raw material for evolutionary innovation and adaptation. With a diminished gene pool, the chances of such beneficial mutations occurring are reduced, hindering the potential for populations to evolve and thrive. It is important to consider the potential consequences of reduced genetic diversity when undertaking genetic modifications. Striking a balance between targeted modifications and preserving overall genetic diversity is crucial to ensure the long-term sustainability and adaptability of species in the face of changing environments. Welcome to the section titled, Long-Term Implications for Species Survival. The reduction of genetic diversity through genetic modifications can have profound consequences for the survival and long-term stability of species. Genetic diversity is a fundamental aspect of species' ability to adapt and respond to changing environments. It provides the necessary raw material for natural selection to act upon, allowing populations to evolve and survive under different conditions. However, if genetic diversity is significantly reduced, the ability of species to cope with environmental challenges may be compromised. In the absence of sufficient genetic diversity, species become more vulnerable to the impacts of environmental changes, such as climate variations or the emergence of new diseases. Limited genetic variations limit the potential for adaptive responses, potentially leading to decreased fitness, reduced reproductive success, or even extinction. Moreover, reduced genetic diversity makes species more susceptible to the negative effects of inbreeding and genetic disorders. Inbreeding can increase the frequency of harmful recessive traits, leading to decreased overall health and reproductive capabilities within populations. This can further diminish the chances of long-term survival and recovery for species. It is important to consider the potential long-term consequences of reduced genetic diversity when engaging in genetic modifications. Protecting and preserving the full spectrum of genetic variation within populations is essential to safeguard species' ability to adapt, thrive, and maintain sustainable populations over time. Welcome to the section titled, Lack of Long-Term Research. Genetic engineering is a relatively new and rapidly evolving field, and as such, our understanding of the long-term consequences of genetic modifications is still limited. While genetic engineering holds great promise, particularly in areas such as healthcare and agriculture, we must recognize the uncertainties and potential risks associated with making permanent changes to the genetic makeup of organisms. One of the main challenges is the lack of long-term research on the effects of genetic modifications. The field of genetic engineering has made significant advancements in recent decades, but the time required to observe and analyze the long-term impacts of these modifications extends beyond our current understanding. New genetic technologies and techniques are continually emerging, making it difficult to predict the full outcomes of genetic modifications over time. The complexity of genetic systems and the potential for interactions between different genes further complicate the understanding of their long-term effects. The limited amount of long-term research means that we may not fully comprehend the unintended consequences of genetic modifications until future generations. It is essential to approach genetic engineering with caution and prioritize thorough and rigorous research to mitigate potential risks and ensure the long-term stability of ecosystems and species. As we continue to explore the possibilities of genetic engineering, it is crucial to invest in comprehensive long-term studies and strengthen our understanding of the potential effects on the environment, biodiversity, and human health. Welcome to the section titled, Unintended Consequences of Genetic Modifications. While genetic engineering holds great potential, there is a possibility of unintended effects that may only become apparent in future generations. Genetic modifications involve altering the DNA of organisms to introduce desired traits or suppress certain characteristics. However, the complexity of genetic interactions and the intricate workings of biological systems make it challenging to anticipate all possible outcomes. One concern is the potential for unintended side effects resulting from genetic modifications. Even with meticulous planning and rigorous testing, there is always a risk of unexpected changes occurring in an organism's genetic makeup. These unintended consequences could impact various aspects, including the organism's health, reproductive capabilities, or interactions with the environment. Additionally, genetic modifications may disrupt existing ecological or evolutionary relationships. For example, if a genetically modified organism is introduced into a natural ecosystem, it could interact in unexpected ways with other species, potentially impacting ecological balance and biodiversity. Furthermore, genetic modifications have the potential to create irreversible changes in populations or species. Once released into the environment, it may be challenging or even impossible to reverse or mitigate the effects of these modifications. As we continue to advance in genetic engineering, it is crucial to proceed with caution and thorough consideration of the potential unintended consequences. Rigorous testing, transparent evaluation, and ongoing monitoring are essential to anticipate and address any unforeseen effects. It is our responsibility to ensure that the benefits of genetic engineering outweigh the potential risks, both for present and future generations. Welcome to the section titled, Potential Risks for Future Generations. The unknown long-term effects of genetic modifications raise concerns about the potential risks that future generations may face. Genetic engineering involves making deliberate changes to the genetic material of organisms, and these modifications can be passed on to subsequent generations. While we may have immediate control over the modifications we make, the full implications and consequences for future populations remain uncertain. One potential risk is the possibility of unintended genetic changes accumulating over time. As modified organisms reproduce and pass on their genetically altered traits, these traits could interact with the existing genetic diversity in unpredictable ways. It may lead to unforeseen genetic mutations, disruptions in gene regulation, or unintended alterations in complex biological pathways. Furthermore, genetic modifications may introduce a genetic burden that future generations have to bear. Traits that may initially seem advantageous may have unforeseen negative effects or become detrimental in changing environments. Future generations may encounter challenges or limitations resulting from genetic modifications made by previous generations. Additionally, the ethical concerns surrounding genetic modifications in the present may continue to have reverberations for future generations. Questions around consent, equity, and access to benefits and potential risks remain relevant as we consider the implications of genetic engineering for the long-term well-being of all individuals and populations. It is crucial to approach genetic engineering with care and a long-term perspective. We must strive for rigorous research, thorough risk assessments, and ongoing monitoring to ensure the ethical and responsible use of genetic modifications for the sake of future generations. Welcome to the section titled, Creating a Designer Baby Culture. The ability to select and modify certain traits in unborn children raises ethical concerns regarding the potential for a society that values designer babies. Genetic engineering techniques present us with the possibility of choosing specific traits in our future children. While this may initially seem appealing, it raises questions about the consequences of treating children as customizable products. One concern is the potential for a stratification of society based on genetic traits. If certain traits are valued and sought after, it could lead to a society where individuals are judged or treated differently based on their genetic makeup. This can perpetuate inequality and prejudice, as those who do not possess the desired genetic traits may be seen as inferior or stigmatized. Moreover, the concept of creating designer babies challenges the notion of natural diversity and the acceptance of individual differences. It raises questions about the intrinsic value of every individual, regardless of their genetic makeup. By placing emphasis on specific traits, we risk devaluing the inherent worth and uniqueness of each person. Additionally, creating a designer baby culture may have unintended consequences on society as a whole. It could influence societal norms and ideals, potentially narrowing our understanding of what it means to be human. This reductionist approach to human diversity may overshadow the richness that comes from embracing and celebrating natural variations. It is essential to engage in thoughtful discussions and ethical considerations surrounding the development and implementation of genetic engineering techniques. By promoting inclusivity, diversity, and respect for each individual, we can navigate the ethical challenges and ensure that the potential benefits of genetic engineering do not come at the expense of our shared humanity. Welcome to the section titled, Devaluation of Natural Diversity. The practice of selecting and modifying certain traits in unborn children raises ethical concerns regarding the potential devaluation of natural diversity. Natural diversity is a cornerstone of our ecosystems and human society. It encompasses variations in physical characteristics, abilities, and genetic traits that make each individual unique. However, the ability to create designer babies may lead to a reduction in the appreciation and acceptance of this natural diversity. By focusing on specific desired traits, there is a risk of promoting homogeneity and a narrow definition of what is considered ideal. This can marginalize individuals who do not conform to these predetermined standards, reinforcing societal pressures to conform and devaluing those who naturally possess different characteristics. In addition, valuing certain traits over others may undermine the inherent worth and dignity of individuals who fall outside the narrowly defined parameters of desirability. It can perpetuate harmful biases and stereotypes, leading to discrimination based on genetic factors and further exacerbating existing inequalities. Natural diversity not only contributes to the rich tapestry of human existence but also plays a vital role in the resilience and adaptability of our species. Genetic variation allows for the capacity to respond to changing environments, resist diseases, and maintain the overall health and survival of populations. As we navigate the ethical considerations of genetic engineering, it is essential to recognize the value and importance of natural diversity. Embracing and celebrating the full spectrum of human variation can lead to a more inclusive and equitable society, where each individual is respected and valued for who they are. Welcome to the section titled, Ethical Considerations for Parental Autonomy and Child's Autonomy. The ability to select and modify traits in unborn children raises important ethical questions surrounding the autonomy of both parents and the future child. On one hand, parents may argue that they have the right to make decisions about their child's genetic makeup. They may prioritize the potential health benefits or desired characteristics they believe will provide their child with a better life. However, this raises concerns about the limits of parental autonomy and the potential for imposing one's own desires or aspirations onto the child. It is crucial to consider the child's autonomy and the right to self-determination. Genetic modifications made without the consent of the future child deny them the opportunity to shape their own identity and make choices about their own genetic heritage. The child may be burdened with genetic traits that they never had a say in, potentially impacting their individuality and sense of self. Moreover, the ethical implications extend beyond individual autonomy to societal implications. The choices made by parents in selecting specific traits may have implications for future generations and the wider community. The potential risks of creating a genetically homogeneous society or perpetuating inequalities based on genetic characteristics raise significant ethical dilemmas. Balancing parental autonomy and the child's autonomy requires a careful consideration of the long-term consequences and ethical responsibilities. It is essential to foster open and transparent dialogue, promote informed decision-making, and ensure that genetic modifications align with fundamental principles of respect for individual autonomy and the well-being of future generations. As we come to the end of our exploration into the ethical ramifications of genetic engineering, it is clear that this field holds immense promise and potential. It has the power to advance medical treatments, increase crop yields, and enhance our understanding of biology. However, with this power comes great responsibility. We have discussed the potential for genetic engineering to be misused for bioweapons, highlighting the need for strict regulations and international cooperation to prevent such catastrophic outcomes. The issue of unequal access to genetic enhancements has raised concerns about perpetuating social inequalities and the importance of ensuring equitable distribution. The loss of genetic diversity, a consequence of excessive genetic modifications, poses risks to the long-term survival of species and ecosystems. It is vital to strike a balance between genetic advancements and preserving our natural diversity. The unknown long-term effects of genetic engineering warrant caution, as unexpected consequences might emerge in future generations. More research and long-term studies are necessary to fully understand the implications. Lastly, the ethical considerations surrounding designer babies raise important questions about societal values, discrimination, and the potential commodification of human life. We must navigate these complex issues with care and empathy. In conclusion, genetic engineering presents us with a world of possibilities, but it also demands our careful consideration and responsible decision-making. As we continue to push the boundaries of science and technology, let us strive for a future where ethical guidelines and thoughtful deliberation guide our actions.